Hello, good afternoon, everyone. Myself, Dr. Rupesh Nakar. I heartily welcome all of you in today's wonderful afternoon. We are hosting second lecture of our lecture number second of lecture series number 3 in 21-22 life science series, which is preparation for NET, GSet, GATE, PhD, and all other examinations which are being held by our state government and other state governments Department of Life Sciences. So I heartily welcome all of you again to today's session and wonderful lecture, which will be on protein and its different types, and it will be taken by our eminent speaker, Dr. Sanket Charola sir, who is from MS University, Vadodara. So our respected Vice Chancellor uh, Pratap Siji Chauhan and respected Registrar Sri Anil Solanki sir, who are constantly motivating us. But in addition to that, today we have with us our media convener and coordinator of life science NET GSET preparation program, along with a whole NET GSET preparation program of all the different subjects, Dr. Ajay Soni sir. So Soni sir is with us. Along with that, from Balasinor Science College, we have with us Satisha Ahmed madam and their two students, Parma Risita and Snehal Patel. I heartily welcome all of you to today's webinar, which is being organized by Sri Govind Guru University, Godhra, for the benefit of students from all over the Gujarat. So, to have a few words to provide blessings, I would request respected Ajay Soni sir to start the program with their best wishes message and blessings to all of us. Soni sir.
ઓકે અવાજ આવે છે બરોબર નાગર સાહેબ
Yes
અવાજ
sir.
આવે છે
Yes sir, આવે છે.
શ્રી ગોવિંદ ગુરુ યુનિવર્સિટીના અમારા સફળ સુકાની ત્રીજી ટર્મ વાઇસ ચાન્સલર બનેલા એવા પ્રોફેસર પ્રતાપસિંહ ચૌહાણ સાહેબ, અમારા ઇન્ચાર્જ રજિસ્ટ્રાર ડોક્ટર અનિલ સોલંકીજી, અમારા સાથી એક્ઝિક્યુટિવ મેમ્બર્સ, એકેડેમિક કાઉન્સિલના મિત્રો, વિવિધ કોલેજના આચાર્યશ્રીઓ, અધ્યાપકશ્રીઓ, વિવિધ કોલેજમાં અને લાઈફ સાયન્સના આજે આ ઓનલાઈન વેબિનારમાં ઉપસ્થિત સૌ સ્ટુડન્ટ્સ મિત્રો, આજના આ લેક્ચરના વિદ્વાન વક્તા જે MS યુનિવર્સિટીથી આપણી વચ્ચે ઉપસ્થિત છે ડોક્ટર સંકેતભાઈ ચારોલા સર, વિવિધ કોલેજમાં પ્રોટીન વિષય ઉપર આપણને વાતચીત કરવાના છે. આપણી વચ્ચે ઉપસ્થિત છે બાલાસિનોરથી સહિષ્ઠા મેડમ અને એમના બે સ્ટુડન્ટ ઈશિતા અને સ્નેહા, એ પણ આપણી વચ્ચે આજે આ કાર્યક્રમમાં ઓનલાઈન કાર્યક્રમમાં ઉપસ્થિત છે. મને જણાવતા વિશેષ આનંદ થાય છે કે ડોક્ટર રૂપેશ નાકર એ વર્ક ઓલિયર પર્સનાલિટી જે સતત કામ કરવું એનો સ્વભાવ છે. વેકેશનના સમયમાં પણ વિદ્યાર્થી માટે સતત ચિંતા કરવી, એને પ્રિપેર કરવા, CSIR માટે, નેટ જીસેટ માટે તૈયાર કરવા, કેરિયર માટે તૈયાર કરવા, એ સતત મંથન અને મથામણ કરતા એવા ડોક્ટર રૂપેશ નાકર એ મારી આ ટીમના મજબૂત સાથી છે અને એમના પ્રયત્નથી ગોવિંદ ગુરુ યુનિવર્સિટી લાઈફ સાયન્સની અંદર આ અમારું 42મું વ્યાખ્યાન છે નેટ સ્લેટ માટેનું. ટોટલ અમારા 227 થી વધારે ઓનલાઈન કાર્યક્રમ થયેલા છે અને આ કાર્યક્રમથી અમારી યુનિવર્સિટી એ મોટી થઈ રહી છે. સ્ટુડન્ટ્સ માટે અમે સતત કામ કરનારા માણસો છે. મને આનંદ છે અને અમારી આખી ટીમને પણ આનંદ છે કે ડોક્ટર રૂપેશ નાકરના પ્રયત્નથી લાઈફ સાયન્સની અંદર દિવસે ને દિવસે કામ વધી રહ્યું છે. તો આ તબક્કે કમ ટુગેધર, think together, work together, teamwork is the key to success ને હું યાદ રાખું છું. અને જેમ મોર એના પીછાને કલગીથી રડિયામણો છે, એમાં નેટ્સ અને જીસેટનો પ્રોગ્રામ એ અમારા કોઓર્ડિનેટર સાથી મિત્રો, અધ્યાપકો, વિવિધ કોલેજના આચાર્યશ્રીઓના સહયોગથી આ એકમાત્ર પ્રોગ્રામ એ વિદ્યાર્થી લક્ષી પ્રોગ્રામ છે અને વિદ્યાર્થીના કેરિયર માટે વેકેશનના સમયના પણ સદુપયોગ કરીને વિદ્યાર્થી જ્ઞાન અર્જન કરતો રહે, પોતાની ઉજ્જવળ કારકિર્દી બનાવતો રહે, એ અમારો એની પાછળનો આશય છે. અને આ બપોરના 3 વાગે આ કેરીની સિઝનમાં રસ ખાઈને બીજા અધ્યાપકો અને આચાર્યશ્રીઓ આરામ કરતા હોય તેવા સમયમાં અમારા સહિષ્ઠા મેડમ, રૂપેશ નાકર અને આજના વક્તા ડોક્ટર સંકેત ચારોલા તે આજે આપણી વચ્ચે ઉપસ્થિત થઈને પોતાના પ્રોટીન વિષય ઉપર વિદ્યાર્થીને માર્ગદર્શન આપવાના છે. સૌના પ્રયત્નને હું બિરદાવું છું. મને વિશેષ આનંદ છે કે આ સેવા યજ્ઞ એ નિઃશુલ્ક ચાલે છે. અમે વિદ્યાર્થી પાસેથી આનો 1 પણ રૂપિયો ચાર્જ કરતા નથી. આ તમામ કાર્યક્રમ એ ગોવિંદ ગુરુ યુનિવર્સિટીની વેબસાઇટ ઉપર, યુટ્યુબ ઉપર અને ફેસબુક પર લાઈવ જોવાય છે. અને જે લોકો આ કાર્યક્રમ અત્યારે નથી સાંભળી રહ્યા એ પણ પાછળથી પણ આ કાર્યક્રમ સાંભળીને પોતાની જ્ઞાન પીપાસાને સંતોષી શકે છે. પોતાને આગળ વધવાની વૃત્તિને, શીખવાની વૃત્તિને આ માધ્યમથી તેઓ એનો લાભ લઈ શકે છે. ગુજરાતના 25,000 થી વધારે વિદ્યાર્થીઓ ને નેક્સ્ટ લેટ માટે અમે મોટિવેટ કર્યા છે અને દિશા દર્શનનું કામ કર્યું છે. અને મને વિશેષ આનંદ છે કે આખો યજ્ઞ એ નિઃશુલ્ક યજ્ઞ છે અને વિદ્યાર્થી લક્ષી છે. કે સમાજના અનેક લોકો, અનેક વિદ્વાન અધ્યાપકો એ અમને જ્યારે જ્યારે અમે બોલાવીએ ત્યારે કાર્યક્રમમાં આવે છે અને પોતાના આવડતનો, પોતાના અનુભવનો, પોતાના જ્ઞાનનો લાભ એ વિદ્યાર્થીને આપી રહ્યા છે. તો આ પ્રયત્નને હું બિરદાવું છું અને લાખ લાખ અભિનંદન ડોક્ટર રૂપેશ નાકર, આલ્ગુની મેડમ અને લાઈફ સાયન્સની સમગ્ર ટીમને. આજે ઈશિતા, સ્નેહા અને સહિષ્ઠા મેડમને પણ હું ખૂબ ખૂબ અભિનંદન આપું છું. આ એક દીપ સે જલા દૂસરા, જલતે દીપ અનેક આ સેવા યજ્ઞ વધારે ને વધારે વિદ્યાર્થીઓને પ્રોત્સાહન આપતો રહે, ઉજ્જવળ કારકિર્દીમાં નિમિત્ત બને, એ જ ભાવ સાથે મારી વાતને વિરામ આપું છું. વંદે માતરમ. જય જય ગરવી ગુજરાત. ભારત માતા કી જય. થેન્ક યુ નાકર સર.
જી. Thank you, respected Ajay Soni sir, who is motivator of all the lecture series which is being organized in different subjects, whether they are arts, commerce, or even in science and life sciences. Ajay Soni sir is one of the wonderful motivator. He keeps motivating all of us. So thank you, sir. Because of you, we are coordinating such series and we are doing such good work for our students. So from Sriji's Science College, today we have with us Sahista Madam, Isita and Sneha. All of you are requested to be connected by muting your mic. We will give you invitation to speak about our program after the lecture. And I also welcome all the students who are watching us live on YouTube and all the other students who are watching us on Facebook as well. So I welcome all of you again. So before we start the lecture, I would like to provide brief biodata of our respected speaker of today's session, who is Dr. Sanket Bhai Charola sir. He is working as an assistant professor in Department of Botany. He has good experience of more than 7 years in teaching and vast experience of more than 10 years in research as well. He has good publication record and have more than 20 to 25 research papers in good international and national level journals. He has also working as a coordinator of net programmation. He is coordinating all the programs related with net in MS University as well. He is one of the wonderful friend of mine. Whenever need is there, he is always there, and he keeps us motivating all the time. So we welcome you again on the occasion of our lecture, sir, and it's a beautiful afternoon here to to see our students who are preparing for series. So welcome you again, sir, and I would like to invite you for starting today's lecture. So welcome, Sanket sir. Now you can start the series.
Thank you, Nakar sir. Thank you for inviting and letting me join this wonderful program started by Sri Govind Guru University, Godhra. I take this opportunity to thank Honorable Vice Chancellor of Govind Guru University, as well as Honorable Registrar Sir, and coordinator and really wonderful orator, our beloved Dr. Soni Sir. And thank you, Nakar Sir, for having me here. I thank one and all who are watching us live and who are trying to learn a little bit about the topic that I'm going to deliver. So let me just tell you that this is going to be the third lecture in a series of concepts about the proteins. We had started with the biomolecules, and then we had landed down to looking at the peptide structure in the last lecture, and we have seen already about the van der Waals forces, about the action of those forces in making Ramchandran plot, and then moving ahead to looking at the secondary structure of the protein. My this particular lecture, that is on the protein structures and types. In this particular lecture, let me just go to the presentation straight. In this particular lecture, I will try to connect to you on different aspects of types of the protein and various aspects of the structures of the protein. Now, looking at the CSIR point of view, this aspect of protein types are quite asked with relation to different types of the proteins, and they are mostly asked in 2 marks questions. So the content that I'm going to deliver will cover large numbers of questions of 2 marks. And in 4 marks question, however, this particular aspect is not directly asked, but it can be related to NMR or X-ray crystallography, or it can go into the dimensions of protein structure identification, or we can say about the mass spectrometry and CD or ORD. So this makes the basis of 4 mark question, but not direct 4 mark questions, and this makes a direct 2 marks question. So I will try to deliver this lecture with keeping this point in mind. So while looking at the studies of this topic, you can refer to wonderful books like Biochemistry by Leninger or Biochemistry by White and White. These are the two major books that I would always tell CSIR aspirants to go for. Those who are studying for master's courses, they can go for Leninger for simplicity. And CSIR candidates can also take Stryer, Lubert Stryer, or Metzler in consideration. Looking at the types of the proteins, so largely we can divide the proteins through its biochemistry point of view into these two major types, that is fibrous protein and the globular protein. Looking at these two major types, that is fibrous and globular proteins, we can go through the fibrous protein. The terminology fibrous makes us understand that something which is of long in length rather than more in its three-dimensional size. So fibrous protein are usually, as the name suggests, they are rod-shaped proteins. They are much longer in length than their other dimensions, and they are insoluble in water, basically providing the strength to the organs. The two major types that we are going to look in the presentation are alpha keratin and collagen. And other type of protein which classification wise different than the fibrous protein are globular protein. These proteins are more compact spherical shaped rather than the rod shaped. And in that nature, they are usually soluble in water. As I've used the terminology usually soluble is because not always soluble. Okay, so that makes sometimes a 2 mark question, one option would be there. Fine. Looking at the first type, that is the fibrous protein. When we look at the fibrous protein, they are highly elongated molecules. Somewhere their size goes up to 2000 to 3000 angstrom in the length, right? And they're mostly along the length of the organ or cell or matrix, etc. Their secondary structures are the dominant structural motif for the fibrous protein, right? So here one more additional difference between the fibrous and the globular is that in globular, however, it will not be just secondary structure which will be the dominant structural motif, but it will be the tertiary structures and the quaternary structures which would make which would make the larger domains of the proteins, right? Major role of the fibrous protein is a role that is there in the protective and connective tissues, and sometimes it is supportive like in skin and tendons. Motive function of the fibrous protein is also known, sometimes that we see in either the muscle or ciliary bodies, wherein this proteins make a movement of different types of cargo in the cell. Looking at the details, structural details of the first protein, that is alpha keratin, that is found in mostly your hair, nail, bone, horns, etc. This particular protein is of again two types. One place wherein alpha keratin will be soft protein, another place wherein this protein will be hard protein. Now, what is the basis of that? Also, we will get into the detail once we will look through the presentation. Alpha keratin, when we take the name, the first thing that hits our mind is alpha, means the nature of this particular structural protein will be in alpha conformation. And whenever we look for the alpha conformation, if you look my previous lecture with relation to the secondary structures of protein, you can definitely recognize that they are helix. The terminology used for alpha keratin is helix of helices. Means it's not a single helix, but it is a helix which is made out of multiple helixes together. So that is why the term used for alpha keratin is helix of helices. It is found in all higher vertebrates. It is mechanically durable and chemically unreactive protein, just like your hair, taking for example, or your nails, right? So they are chemically unreactive and mechanically they are quite durable. Alpha keratin occurs in mammals. However, if you look for the birds or reptiles, there you will find beta keratin. There is a little difference between the alpha and beta form of keratin. Mostly speaking, keratin are rich in cysteine residues. You know about the cysteine, one of the sulfur-containing amino acid, which takes part in forming disulfide bonds in the protein. So looking at cysteine-rich alpha keratin, we can find out that they are rich with disulfide bonds. When a protein is rich in disulfide bonds, if you use mercaptans like beta mercaptithanol, when you use mercaptans, you can simply break these disulfide bonds and can rearrange disulfide bonds once you again provide the reducing environment. This particular statement is a basis of the hair treatment wherein you undergo the curling of the hair, right? Treating the hair first with the beta mercaptan, and then after then allow it for the disulfides to refold, and they will have a curls, right? Another basis of, as I have used hard keratin and soft keratin, it is again the concept which is because of the presence of cysteine. If you have huge amount of disulfide bonds, the protein would be hard protein, and if it is less, vice versa, the protein will be softer one. There are two major type of the polypeptides of alpha keratin that we see. One is acidic and another is basic, based on which we can either call them as type 1 keratin or type 2 keratin. Keratin, if you look for the hair, this is for example the structure of keratin. We will find that there are many dead cells, right? And the cells which are composed of nearly 60% of the keratin protein. These keratin proteins are of a two larger structural dimensions. One we call it as macrofibril, which are in turn made up of many microfibrils. Macrofibrils are always going to be arranged lengthwise the length of the organ. Means length of the organ as in, if you can take example of hair, the towards the length of the hair, macrofibril will be arranged. If you look at the dimensions, macrofibril is of 2000 angstrom and microfibril in contrast is 80 angstrom. So you can just quickly count that there must be nearly 300 microfibrils which can make a large macrofibril. And so many such macrofibrils would take part in making a single cell of the hair. Looking at the structure of alpha keratin, quite complex one. If you look at the structure of alpha keratin, it is simply of a right-handed alpha helix. Okay, this is the first thing that we need to remember, right-handed alpha helix. You can know that alpha helix can be of two types, right-handed or left-handed. This is made up of right-handed alpha helix, in which there will be many alpha helix, as I had told you, which comes up together to make it is called as helix of helices. This particular structure is a coiled coil structure of a many alpha helices put together. For example, in the first inset figure over here, this particular figure is representing a first dimer of a alpha keratin protein. So you can look for, these are the two polypeptides which are intervened with the formation of right-handedness. So it is a helix which is of right-handed, which in turn is a helix. So this yellow color structure that you are seeing is one alpha helix, and this gray color structure that you are looking at is another alpha helix. So two such alpha helix of keratin are intervening to make helix of helices. That is called as coiled coil structure. Coiled coil is a consequence of a primary structure. Primary structure that is a typically nothing but a simply peptide bonds. So interesting about the peptide bonds in case of alpha keratin, which leads to such secondary structure, is that there is a central almost 310 residues of one polypeptide chain, each of type 1 and type 2 alpha keratins associate in a dimeric coil. So you can just understand that this is whole is a 450 angstrom. Out of this 450 angstrom structure, there will be some central part of nearly 310 angstrom 310 residues. These 310 residues of a polypeptide are going to associate for this dimerization, okay, which is comprising of 450 angstrom. This particular structure form a protofilament. So many of such dimers would join end to end like the N terminal. So you can see these are N terminal heads and C terminal tails. They will join head to tail arrangement. You can see this is the head and this is tail. The other one is in head here. So it is a head to tail arrangement. It will form a staggered head to tail structure which makes a protofilament, right? So these are the two staggered anti-parallel rows of associated head to tail coiled coils making a single protofilament. Two such protofilaments would make one particular protofibril. As you can see here, the terminology is written protofibril. So two such protofilaments, like one circle over here, making one protofibril, and four such protofibrils would make a larger organization, microfibril. And as I have told you in the previous slide, there are 300 such microfibrils can make one microfibril. So this is the organization, right? Now, what made this nearly 310 residues to come together to form such a coiled coil structure? That's the question. So looking at those 310 residues of a single polypeptide chain, you will find that there is a heptad pseudo repeats. What do you mean by pseudo repeat? That in this 310 residues, 7 amino acids will be keep on repeating for multiple times. As you can see, the repeats is of 7 residue. For example, taking here A, B, C, D, E, F, and G, 7 residues. With A and D, okay, the first one and the fourth one would be a non-polar residues, okay, predominantly non-polar residues, A and D. If you remember the secondary structure of alpha helix is having 3.6 residues per turn, okay. Like if you take consideration of this particular structure first round, it is 1 turn, if you can say, then consider A, then B, then C and D, right? So 3.6 residues would be there in one single turn, okay. Rather than 3.6, in case of alpha keratin, it is 3.5 residues. Okay. So what will occur is every time 3.5 residues you repeat, A and D would come on the same side, on one single side of this helix. So consider this is a cylinder going inside of the screen, right? and looking at A and D being always on one side. So you keep on repeating these haptards and you will find that there is a sequence of A and D keep on coming on one side. Fine. So this make a hydrophobic, because this A and D are hydrophobic amino acids, this make a hydrophobic strip on just one side of the cylinder. This is one side of the cylinder, right? Now you can consider that there were two such alpha helices put together to make a dimer. So consider this is one alpha helix and this is another alpha helix. It has A, B, C, D, F, G. This has A prime, B prime, C prime, D prime, E prime, F prime, and G prime. Considering A prime and D prime coming together on one single side. So this is also hydrophobic and this as well hydrophobic. They both would interact being hydrophobic because there is a hydrophobic interaction, non-polar interaction. This makes the basis of a coiled coil conformation. Okay, so this is the first information about the interesting phenomenon of how coiled coil structures are made. You can just revise that two polypeptides, one is type 1 and another is type 2. They come together, they make a coiled coil structure to make a dimer. Dimer would make a fibril, then the fibril will make a protofibril, then protofibril to go on to microfibril, and microfibril to macrofibril. This is the organization of keratin structures. Structurally, hydrophobic interactions are one that you need to remember, and right-handedness of the alpha helix that you must remember. Comparison of two types of keratin, one is known as alpha keratin and another is known as beta keratin. Alpha keratin is mostly tough, insoluble type of keratin. However, beta keratin is a soft and flexible type of keratin. Alpha keratin has alpha helix conformation. However, the same alpha helix type of conformation, when we look at the beta keratin, it becomes an extended chain conformation having hydrogen bonding just like beta plated sheets. So alpha keratin is of alpha helix, beta keratin is mostly of beta pleated sheet. So basic unit in alpha keratin is protofibril, in beta keratin it is beta pleated sheets. Alpha keratin has cysteine as important amino acid. As I told you, more amount of cysteine means more amount of disulfide bonds, means more the tougher the protein is. In contrast, alanine and glycine are two important amino acids in beta carotene, which gives more beta pleated sheet of arrangement to beta carotene. Devoid of cysteine, or rather we can say less amount of cysteine in beta carotene, makes them more softer and flexible. Looking at the next type of structural protein or fibrous protein, we must say, that is collagen. Rather than alpha keratin, collagen is a triple helical structure. Keratin was double helical, this is triple helical. In this type, there are three different helices put together forming a single collagen molecule, that is what is a basis of collagen structure. It occurs in all multicellular animal in contrast to keratin, and it is the most abundant protein of vertebrates. Of a cell, nearly 60% of the protein in the structural components you can attribute it to the collagen. So it's a quite a huge amount. Vertebrates has nearly 28 distinct collagen type. You can name like type 1, type 2, type 3 up to type 28 different types of collagen are there. Many times it's type 5A, type 5B, etc. etc. We don't need to get into the details of them. However, you can look for the chain composition of each of them. Like for example, in type 1, you have one particular alpha 1 molecule which is occurring two times and another is alpha 2 molecule. However, in contrast, type 2 has all alpha 1, right, is a triplet, and type 3 has again triplet of all type 3 types of the chain composition, right? So each of these types are made up of nearly 60 genes on the chromosomes. Fine. Looking at the type 1 collagen, it is composed of three different polypeptide chains, that is two of type alpha 1 and one of alpha 2. It is predominantly found in skin, bone, tendon, blood vessels and cornea. which attributes to the transparency of the cornea. Of course, we must say, if rather in cornea, if you get the if you get the lesion on the cornea and you find the fibroblastic activity on the cornea, you will find that the type 2 collagen comes up, and then the cornea becomes fibrous and it will become hazy. Okay, so the type of collagen is extremely important for the particular organ to keep its normal functioning. For example, type 2 is found in cartilage or in vertebral disc, and type 3 is mostly seen in blood vessels and fetal skin. Looking at the structure of type 1 collagen, as I've already mentioned, it is made up of 3 polypeptide chains. An aggregate molecular mass of this is 285 kilodalton. It's a rod-like protein with a length of nearly 3000 angstrom, and you can consider this to be a 300 nanometer, means nearly zero point 0.3 micrometer, right? And a width of nearly 14 angstrom, right? So this is the dimensions of a collagen. So you can consider it's an extremely thin, and the fundamental unit of collagen means the triple helical structure. The first triple helical cable of collagen is known as tropocollagen. That's a fundamental unit. And so many such tropocollagen would come together to make a higher order organization of a collagen structure. Looking at the structure of a tropocollagen means the triplet structure. How does that comes up? This particular collagen and keratin are most important questions for many of the exams. Largely speaking, these questions are asked multiple times. Nearly one third of the residues of the collagen are glycine residues. Okay, one of the most predominant amino acid found in collagen. And you also already know about the glycine and proline, that they are helix breakers, right? Another 15% to 30% of the residues are proline and 4-hydroxychlorolyl, right? These residues. So apart from this 15 to 30%, there is smaller quantity of 3-hydroxyprolyl in contrast to 4-hydroxyprolyl, as well as 5-hydroxylysyl. Okay, hydroxyprolyl are written as HIP and hydroxylysyl as HIL, residues in smaller quantities, right? Looking at the structure of each of this, you can just find that there is very little difference between 4-hydroxyprolyl or 3-hydroxyprolyl. There is an attachment of hydroxyl residues at either fourth position or third position in proline. However, 5-hydroxylysyl is completely different altogether, and you can see that there is an alpha amino acid, alpha amino group with the fifth carbon having one hydroxyl group. These are all modified proline residues as well as lysine residues. Now we can consider that how the triple helical structure is formed. So you can say that the hydroxylated residues appear after the collagen polypeptides are synthesized. So first, as you know, that in translation of protein, the molecules of a tRNA would bring only proline but not hydroxyproline, and they will make a protein. After the proteins are made, then you will find that the hydroxyl residues are added after the collagen polypeptide is synthesized. So this particular residues which have got hydroxylated are called as hydroxyprolide residues, and this reaction is catalyzed by an enzyme known as prolyl hydroxylase. Okay, prolyl hydroxylase, however, require vitamin C, which makes the basis of understanding why in scurvy there is a problem with the skin lesions. So scurvy is a disease from a very early classes. You have known that it is a disease which is caused by vitamin C deficiency, and because of the deficiency of vitamin C, prolyl hydroxylase would not be able to function to its full efficiency, and so it will not be able to form hydroxyproline residues in collagen, and hence the collagen which is synthesized in such an individual will be defective, leading to the skin lesions in scurvy patients. Hydroxyprolyl residues confer stability on the collagen by intramolecular hydrogen bonds that involve bridging the water molecules. So this is the basis of why integrity of collagen requires hydroxyproline, and without that the collagen will not be in its tough form. Looking at how the triple helical structures are formed, so as I've already told you, glycine is nearly major amino acid found in collagen at the first position, and then there is a triplet which followed by glycine is X and Y. At the presence of X, mostly it is proline residues that can be found, and at the position of Y, it can be hydroxyproline. You can see this is a simple structure of a protein that has been shown that at every first position of this triplet, it is remarkably occurrence of glycine. Second position largely is proline residues, and the third position largely it is hydroxyproline residues. Collagen's three polypeptide chains, which individually resembles polyproline 2 helices. I had talked about polyproline 2 helices in my previous class of secondary structures. It very well resembles this polyproline 2 helix. They are all parallel and wind around each other with a gentle right-handedness in a rope-like twist to form a triple helical structure. So you can see this is first peptide unit, this is the second, and this is the third, and you can see that all these three can wound around each other to make a right-handed rope-like structure of a tropocollagen. How hydrogen bonds takes part in collagen? So looking at gly X and Y residues, they are always staggered, right? They are always staggered in the polypeptide chain so that always this gly X and Y residue can form the three chains at a similar level. So you can see that one is gly, okay? There is a group of glycine, there is a NH group of the glycine. It gets connected or hydrogen bonded with the carbonyl group of an adjacent polypeptides proline residue. So you can see this is the carbonyl group of chain 2 which binds with the NH group, amino group of the glycine residue. And subsequently, this kind of a reaction would go on to join to the third chain or to the fourth chain, etc. etc. So this is the pattern of hydrogen bond that occurs among the neighboring chains in the collagen. There is a disease, however, known as letharism that is caused by regular ingestion of seeds from the sweet pea, that is Lathyrus odoratus, having ample amount of beta aminopropionitrile. Beta aminopropionitrile is a particular component, particular compound that can cause abnormalities of joints, bones, as well as blood vessels. Now, what is the basis of this letharism? So, basis of letharism can be pointed out to a very specific enzyme known as lysyl oxidase. Lysyl oxidase is a copper containing enzyme. Its many orthologs are present in an individual because it's an important enzyme which can convert lysine residues to those of the aldehyde lysine. This particular lysyl oxidase converting lysine residues to lysine can link up to 4 side chains by the covalent bonds. So up to 4 side chains of the polypeptide can join via lysyl oxidase to form a very compact structure of the collagen. Now, collagen not cross-link like keratin, as keratin have a cysteine, they can cross-link through disulfide. So in contrast to keratin, collagen rather cross-link to stay in place to increase its strength by the lysyl oxidase. Typically, this beta aminopropionitrile is an inhibitor, or we can say inactivator of lysyl oxidase by the covalent binding to the enzyme. So if you consume a lot of amount of Lathyrus odoratus giving rise to beta aminopropionitrile which can inhibit lysyl oxidase can in turn loosen the collagen by not allowing collagen molecules to get cross-linked, right? So this is the basis of letharism. And apart from letharism, there are epidermobullosa syndrome, right? That also comes up due to the collagen disorder with one or the other enzymes. So collagen is quite an important enzyme, quite an important structural protein required for the proper functioning of many tissues. If you look at the different types of tissues like tendon, skin, cartilage, and cornea, how the collagen is arranged, so you can see there are various types of arrangement like parallel bundles or otherwise layered fibrils at a specific angle, or cartilage having no distinct arrangement, or cornea to remain transparent have a planar sheets which are stacked crossways so that it can minimize the scattering of light, giving the cornea completely transparent appearance. Looking at the third important structural protein, that is elastin. This particular protein is a connective tissue protein, and it is known for its extensibility and elasticity. It is found largely as elastic fibers in ligaments, lungs, and larger arteries. This particular protein is with a partial hydroxylation of protein can get converted into a mature polypeptide known as tropoelastin, which is secreted from the cell in extracellular matrix. Once this tropoelastin is sent to the extracellular matrix in its hydroxyproline form, a few of the few of the hydroxyprotein residues would undergo, few of the lysine residues would undergo oxidative deamination to convert them into aldehyde by enzyme known as lysyl oxidase. Okay, so those few of the lysine residues which gets converted to aldehyde by lysyl oxidase would not have terminal amino group. What will happen then? Then condensation of three such aldehyde containing lysine, right? Or we can say lysyl derived aldehydes. So you can see this is one such aldehyde molecules which is deaminated. This is another such molecule, and this is the third such molecule. All these three molecules would require one more joining hand of a proper lysine containing polypeptide. So four polypeptides coming together, of which three polypeptides lysine residues have been converted to aldehyde, one is still intact. This structure of a complex condensation of 3 plus 1 residues, which is known as a tetra-functional crosslink, is known as desmosins. Okay, so if you break the elastin molecule, this desmosins would come out of the peptide because they are much stronger than the peptide bond in that case, and it is a highly insoluble molecule. So in such case, if you treat the molecules with an enzyme known as elastase, elastase is the enzyme which will break down all other peptide units, only desmosins would remain in the solution. Then if you count the amount of desmosin, you can count the amount of elastin. So desmosin is hence known as the indicator or biomarker of the elastate degradation. Okay, so that's all about the fibrous protein. That's all about the fibrous protein. Now moving to the next type, that is globular proteins. When we look at the globular proteins, the first thing that comes up to our mind is a protein which is more like a globular in its shape. So you can ask, is that all the proteins do not look random? No, answer is that they are chiefly of two types. Either they are globular or they are fibrous. However, few more modern types have been added to it, but classically for CSIR purpose, we will remain sticking to the two major types. Globular proteins comprise a highly diverse group of protein, and in the native state, they exist as a compact spheroidal molecule. As you can see in the inset figure over here, which is the first globular elucidated protein of a sperm whale myoglobin, you can see in its outer periphery it appears more like a three-dimensional globular structure. It may contain sometimes alpha helices alone, sometimes beta sheet alone, or majority of the time they contain alpha helix and beta sheet both. Fine. Now they are mostly in their mostly they are in their tertiary or quaternary protein structures. So I had mentioned, if you remember, during the fibrous protein discussion, that it is only and only secondary structure. This is the tertiary or even the quaternary structure. So the discussion becomes quite difficult for us to understand the moment we reach towards the tertiary or the quaternary structure, because the complexity of the structures would increase. But if in your mind alpha helix and beta sheets are very clear, you don't have much of the trouble understanding the globular proteins. Some of the characteristic features of the globular proteins are that protein secondary structures like alpha or beta in in the tertiary structure of the globular protein, they are folded for more special disposition of the side chains. Wherever the side chains were lagging, just like that, they were just halted in the cell. However, in the case of globular proteins, you can make or you can put them at work, right? So that's the beauty of the what do you say, alpha and beta been folded with the special disposition of the side chains. Looking at first such example is a myoglobin, which consists only of alpha helices, which is spanned by short connecting links that have coil conformation. So you have many alpha helices, and they are joined just by coiled conformation, right? That is called as myoglobin. Or you can look for the concanevalin A, which is largely proportion of beta sheets and having almost no alpha helices. You can also have a mixture of both. Example is like carbonic anhydrase or carboxypeptidase A or triphosphate isomerase, wherein most of the proteins have a significant amount of both the types of the secondary structure, having on an average 31% alpha helix and 28% beta braided sheet, along with all the other necessary twists and turns and motifs and folds and domains. Everything will be there apart from these two major components. Comparing the two major discussions that we had done before, that is the all beta sheets, right? The first one, that was the concanevalin A, wherein all beta sheets are present. You can see that with the arrow there is only beta sheets. Whenever you find the structure, if you don't understand about the diagram, cartoon diagram of protein structures, always remember, as I am drawing over here, whenever it is showing with the arrow, that means it is a beta sheet, and whenever you can see that it is shown as a spiral, then we can say that it is nothing but a alpha helix, right? So here in the case of concanevalin A, it is simply having beta sheets, so it's quite evident. However, in case of this is carboxypeptidase A, you can see that they just simply do not have beta helix, they also have the, what do you call it, as the alpha helixes. Looking at the side chains of the globular protein. So in globular proteins, there are certain rules that we need to remember while working with the globular proteins. The primary structure of globular proteins are generally the ones which they lack repeating or pseudo repeating sequences. So repeating or pseudo repeating sequences, like we have seen that A, B, C, D, E, F and G, making the polarity in such a way that all the hydrophobic amino acids come on a single side. Such kind of a primary structure advantage is not available for the globular proteins. So in that case, amino acids, however, are spatially distributed according to the polarity of the amino acids. Now this particular 6 lines that are highlighted with the green color over here, These 6 lines are largely important for 2 marks question. Many times these questions are asked. So you can see that non-polar residues. Who are non-polar amino acid? Which are polar amino acids? Which are aromatic amino acids? Which are sulfur-containing amino acids? Right? Which are small molecule amino acid? These are all questions you must remember always. So non-polar residues like valine, leucine, isoleucine, methionine, and phenylalanine are largely occurring on the interior of a protein out of control with the aqueous environment. In that condition, what will happen? Whatever numbers of the amino acids that you are seeing in gray color over here, these are all largely what we can say is either valine, leucine, isoleucine, methionine, or phenylalanine. Because they remain buried inside, they always stay away from the contact of aqueous solutes. Okay, so aqueous solvent do not solubilize them, so they always try to remain into the colony, into the characteristic holding each of these amino acids together by hydrophobic interaction. Next one is the charged polar residues, like example arginine, histidine, lysine, aspartic acid, and glutamic acid. All of these particular amino acids are largely located on the surface because they poses an environment which is charged non-polar environment. Next one is the charged uncharged polar groups. Uncharged polar groups example are serine, threonine, asparagine, glutamine, tyrosine, and tryptophan. They are usually on the protein surface. Okay, they are uncharged and polar, but frequently they occur in the interior of the molecule. Whenever a non-polar wants to remain interior into the molecule, they will stay very minimal in contact with water, and so that repulsion of oil water interface, which requires energy to put both of them together, is not required in case of the protein structures. Looking at the domains of a large polypeptide, polypeptides that consist of more than 2000 residues usually fold into two or more globular clusters known as the domains. So if you can see here the term domain, it is largely, we can see from the description, that it is two or more globular structures. These two or more globular structures which or clusters which make a single domain can often give these proteins a bi or multilobal appearance. As you can see, this is the structure of glyceraldehyde 3-phosphate dehydrogenase, the name that you call it as GAPDH, having NAD plus as a cofactor. GAPDH has NAD plus as a cofactor. So domains, they are present in this figure, of which the orange color has predominantly only and only presence of beta sheets. However, in case of the lower one, it is only presence of alpha sheet, right? So this is a alpha helix, rather we should say. So this can also occur. Domains, however, are often a specific function, and here in the gap DH, for example, the specific function attributed to this domain is NAD binding. So what are we trying to make a point over here? When we look at the primary structure, it's a simple chain of amino acid. When we look at the secondary structure, it can be either wounded in form of alpha helix or beta pleated sheet. When we look at the larger organization of such alpha helix and beta sheets, they can either alternate in the same polypeptide, or sometimes they, rather than alternating, their one domain, one part remains only what do you say alpha helix dominant, other part becomes completely beta sheet dependent, and they both again come together and interact with the presence of ligand. So this is typically an example. Moving to the next discussion, that is a super secondary structure. So while looking at the looking at the fibrous and global proteins, large numbers of unusual structures of the protein that we find in the plants, they are discussed in all this, rather plants I must say, in all the organisms. Now, considering just alpha helix or beta sheet, if you can put together both of them and you try various options, right? Then you will be able to come to a conclusive option that is super exclusive, secondary exclusive structures. For example, the first one is beta alpha beta motif. Looking at the beta alpha beta motif, as you can see in the figure over here, the just a minute. As you can see in this figure, there's a red color beta sheet, then there's a green color alpha sheet, and there's again a beta sheet. Next one can be beta hairpin motif. This particular hairpin, because it's going to be used as a hairpin, you can see just the U pin or hairpin that you are seeing in regular life. Very similar structure of the protein, wherein it is made up of only beta sheet but anti-parallel beta sheet. So the joining will be like C to N and N to C and again C to N or something like that. In that case, this anti-parallel beta sheet that is formed by the sequential segment of polypeptide chain that are connected by relatively tight reverse turns would look something like this. Is the second figure. And the third figure is of alpha alpha motif, wherein you have a two alpha helices put together, just like what we have seen a beta beta motif. This also makes a alpha alpha motif with a little turn involved in between, wherein these two alpha helices are going to be packed against each other with their axis, which is usually inclined. What is so important about inclined axis, which I did not take in alpha carotene, is little bit inclined axis of the two helices, because these two helices cannot make a disulfide bonds. So what will happen? Helices, whenever you make, they will have a grooves in them. So if you just make little twisted, little inclined, those grooves are more open and accessible for another such helix to come and interact. And in that case, they both will make a tighter helix, right? There is the beauty of super secondary structure. Moving further with the super secondary structure, you have a Greek key motif, which is very easy to understand that they have a four anti-parallel beta sheets, and this four anti-parallel like 1, 2, 3, and 4. This four anti-parallel beta sheet can simply can be folded by 10 different possible ways. What is the 10 different possible ways? For example, this is a flexible hinge, then you can take 2 next to 1 and 3 also next to 2, so this both will come down. So similarly, there are 10 different ways for rearranging this 4 anti-parallel beta sheet, out of which the one that we are seeing in the figure over here, having 1, 2, 3, and lastly the 4 being quite away, this particular structure is known as Greek key motif. It looks very much like some Greek key, and so this is known as a Greek key motif, which is the most abundant in proteins of known structure. Fine. Next one, when we say that there's a fold, so what do you mean by fold? So group of motif completes in overlapping and non-overlapping ways to form the tertiary structure of a domain, right? So groups of various kinds of motifs they combine together in overlapping and non-overlapping ways. So sometimes, for example, this is the group of, for example, this is one single motif, right? That is a Greek key motif. You have another motif, for example, that is alpha alpha motif, alpha helix motif. Then both of this, they overlap each other, or sometimes non-overlapping but put one next to the other, right? two Greek key motif just placed one next to the other. Such kind of structures, mix and match of various such super secondary structure of out of this any of the four motif, if they are put together, they make a domain. Okay, so they make a they make a fold, which is known as, which in larger terminology, so many such folds can lead to a domain. Okay, so this is the basis of a super secondary structure. So looking at the identity of the domains, I have kept the discussion little truncated to the domain, is because a domain is not so much in detail asked in any types of exam, because this becomes quite little more complex. However, the terminologies like beta barrel, if somebody asks you, then you must be able to tell them that what is exactly a beta barrel. Or for example, a 7 pass transmembrane protein, that is known as G protein, right? It is 7 times passing through the plasma membrane. Then you must be able to tell, just looking at the domain, what kind of domain they are, right? So looking at the types of the domain, sometimes they are simply domains made out of only alpha helices. Sometimes they are made up simply out of beta helices. Example is this particular domain, which is of immunoglobulin domain. You know about the structures of immunoglobulins, IgG, IgA, IgE, etc. This ones wherein you will find that there are presence of many of the beta pleated sheets put together to make a beta domain, right? And what is so important of the domain, that they will not simply be made out of beta sheets, but they will also have loops and turns and twists, and there will be parallel, sometimes there will be anti-parallel. That is the beauty of this beta domain. Next one, in contrast to the beta domain, is a alpha beta domain, okay, which is a name of carboxypeptidase. This one is a protein wherein you see that there is a beta pleated sheets, and various beta pleated sheets. Apart from beta pleated sheets, you will also find a presence of a alpha helices here in between. That is called as alpha beta domain. Next one is known as alpha beta barrel. Okay. Now, one thing that I forgot to tell you was with relation to the beta domain, that a little more complexity of the beta domain that we see than the previous slide here, that has been shown as a Greek key motif. Here what will happen is four sequential anti-parallel beta pleated sheets. If you put together, there it was only two such sheets. Here there are four different, like you can see, four different colors: red, orange, sky blue, and the blue. And in this particular, all these four, if you just simply arrange them as shown in the figure over here, it would be making a structure which looks like this, which looks like a roll. Okay, and because of that reason, it is known as Swiss roll model, Swiss roll structure. Okay, so Swiss roll structure is also little typical question that is asked, which is made out of 8 anti-parallel beta sheets, which makes near like a four different anti-parallel sheets, right, put together. That is what is four different types of anti-parallel beta sheets put together to form a domain which is similar to Swiss roll. Okay, or they say the simply a cake, Swiss cake, Swiss roll sort of a model. Next one is the alpha beta barrel, in which, as the name suggests, I will just go to the next slide to show you the alpha beta barrel. This is typical of a protein wherein there is a presence of a beta sheet with the arrow, and there is a alpha. Then again it alternates with the beta, then alternates with alpha. So alpha and beta alternates in this particular structure to make a alpha beta domain. In this condition, it's so beautifully arranged that, as you can see, all the beta sheets would be arranged in the middle of the protein, and all the alpha sheets will be arranged on the periphery of the proteins, right? So this is a very typical arrangements. Proteins would take up varieties of such different structures, and it is known that nearly 1200 of the folds, as I had discussed in the previous slide, they are already known. Okay, different 400 types of folds, there are 1200, sorry, my mistake, types of folds are already known to us. So you can just understand the complexity of the folding, that looking at the complexity of the folding, you can devise so many types of the tertiary structure, which makes the basis of globular proteins, right? So globular proteins are the largely amount of proteins which are present either as a transmembrane protein structure, either as a functional proteins, either as a, ligand binding proteins, enzymes, coenzymes, etc., etc., various type. Apart from just simply structural proteins, globular proteins plays major role in the cellular activities in a normal cell. Lastly, you had a open beta sheets. In open beta sheets, rather than remaining beta sheets in a more compact fashion, they remain more open in a protein, and that is what is called as the open beta sheet. However, not put the figure of that, but as I told you that knowing so much in detail about each and every one is not so necessary. Alpha beta barrel is essential for us to know for simply one more reason, that what is the importance that is conferred by this barrel. Okay, even the Swiss roll model. So considering, I will just take a help of a pen. Considering a globular protein, okay, you can consider a globular protein either as a simple solid, completely what you can say, completely sphere without any space in between, right? A compact sphere. So considering a compact sphere which has a nearly compaction ratio of zero point 0.8, in contrast to that, globular proteins has a ratio of 0.76. Okay, and lesser to that, you will see more open structures like oil droplet. It will have even lesser than 0.7. So considering that that the globular proteins are not hollow proteins, their core, their center is filled. Okay, they are not hollow. This is the first thing you must remember for globular proteins, because this kind of question is asked. But when we looked at these structures, globular proteins are seen with some space in between. Like you can see this, that you can see that there is a space here, there is a space here, there is a space everywhere around. Okay, so you can consider that there is a space everywhere, but this space, looking at the compaction ratio, has been worked out that this space is not sufficient space for any of the molecule to get through, pass through them. So how this space is filled? So we know that in globular protein, the internal core is filled with hydrophobic amino acid, and the external is hydrophilic amino acid, which remains in contact with aqueous environment. That is very well understood. The amino acids, which I will lead you to the slide wherein we had discussed about the uncharged polar amino acid. Whenever we look for this amino acid, they can usually be present on the surface, and frequently they also occur at interior of the molecule. So what will happen? There will be some functional amino acids which may make hydrogen bonding. What will happen? Any such amino acids which are present in the interior of or the core of the globular protein, they will make all possible hydrogen bonds so as to completely nullify any more hydrogen bonding possibility inside the core. So with all possible hydrogen bond, now no charged amino acids will be left, no charged group will be left in the core, making the core apparently neutral or hydrophobic. This is the first thing, so that the core is extremely compact in its nature. Okay. Now, when we consider about the transmembrane protein, so when you consider, let's say, this protein itself as a transmembrane protein, in that case, with the transmembrane protein, its periphery should be connected with the hydrophobic character of the fatty acids of lipid molecule of phospholipid molecules. So when we consider that if this molecule is a globular protein, and we by the definition we saw that all the non-polar amino acids are buried inside, all the aqueous or the amino acids which are reactive to the aqueous environment are outside, then this does not apply the same in this case. In that condition, what will happen is that all the non-polar amino acids will be present outside interacting with the phospholipid core, and the internal will be polar residues, so that this particular barrel would be able to allow the free passage of some molecules like metal ions, salts, or we can also say sometimes small molecules like glucose, etc., can enter through this particular pore, right? In that case, this molecule would be a barrel rather than simply a globular protein. So this is what in the Swiss roll model, if you look through, all these connecting loops are not letting the barrel to function as a passage of any molecules. It does not allow the way that I am drawing the diagram. It does not allow any free passage. So we can consider Swiss roll as a compact molecule. But when we look at the barrel, in barrel there is a presence of gap in the middle which lets the free passage of molecules in and around. Fine. So there's a understanding of the compaction of a globular protein and the arrangement of amino acid either on the core or in the surface. Looking at the structure that I've discussed in today's class, there is a specific classification. Those who are looking for biochemistry entrances, they ask about this particular classification. It is given by scope, or it is known as scope, that is structural classification of protein. Okay, that is a scope, and the website I have already listed over here. You can just go through it, and there the classification are given on the basis of two major types. One is a structural class of the protein, and next is the type of the protein. In today's particular discussion, I have focused on only these two particular part. One was the type of the protein, in which we only saw two major type important for CSIR, that is globular protein and fibrous protein. But I've already told you that you must remember some more additional type like membrane proteins and non-globular intrinsically unstructured protein. As I told you, globular proteins having core being non-polar, membrane proteins will have the surface being non-polar, and the non-globular intrinsically unstructured proteins have no specific structure. Their core is hydrophilic, their surface is hydrophilic, but the core is largely unstructured. None of the motifs or folds that we have seen in the previous class would apply to this. So if you go through the classification, the criteria of classification, if you go either through structural class or through protein type, it will go on. Next are the types of the fold. As I've told you, there are nearly 1200 different types of fold are already known. So the classification will be given by folds. If you click on each of the fold, you will find there is a superfamily. If you click on the superfamily, any of the superfamily of your interest, then it will be classified as family of proteins, and family of protein will be in turn given the classification based on the domain, right? So there are few domain but so many fold that you must remember. Okay, and based on that this classification are given. That's known as structural classification of proteins. Same can also occur, same hierarchy is also there in the protein type also. Any of the protein types you can click and find out. So that's all with relation to the delivery that I wanted to do. I'm finishing it within an hour as given. And so just looking at the lastly one question that I have put up, that is which of is a CSIR question, and which of the following is the most appropriate statement regarding folded proteins. You can see charged amino acids side chains are always buried inside the core of the protein, or charged amino acid side chains are seldom, only sometimes buried. Non-polar amino acid side chains are seldom buried, or tyrosine residues are always buried inside. Looking at this question, you can just go to my discussion about this particular names of the amino acid, and you must remember that the non-polar residues should always remain buried in the interior core of the protein molecule. So you'll get the answer, right? So with this, I would like to close my today's session on protein types and the structure, wherein I have discussed only about the fibrous and globular proteins, and the structural classification. I've talked about the folds and domains in large. And apart from this, when we go to the quaternary structure, okay, like when we talk about the quaternary structure of hemoglobin, myoglobin, and then we enter into the cooperation of each of this particular domain and its function, probably I'll take it up into the next particular class. And moving forward from there, we will try to understand the oxygen saturation curve, and we will try to understand the cooperativity models of the proteins. And then we are firm to understand about the enzymes in detail, right? So that's all for this particular session. I take this opportunity to thank one and all the administrators of Shri Gobind Guru University, Godhra, and special thanks and regards to coordinator of the program, Dr. Ajay Soni sir, and Dr. Rupesh Nakar sir, for continuous motivation to the students through this wonderful program. And it's live, so many students can watch this at their leisure. I would like to take the queries from you, those who are at present joining this session through the web portal. And otherwise, you can write to me on my email I have put up over here, and I will be very happy to answer your queries. You can go through CSIR 2 marks question, very direct questions will be there through the discussions that I have given. And as I've told you, domains, folds, etc. are important components, along with my previous session, of the propensities of amino acids for making either helix or beta sheet, will be largely helping you out in determination of protein structure, as well as in looking at the CDORD, X-ray, NMR, and mass spectrometry based questions. Okay, so thank you all. Thank you one and all. I would like to invite queries from your side. Thank you.
Uh, Thank you, dear Sanket sir, for delivering such a nice and in-depth lecture. And we truly enjoy your lecture. You described about fibrous and globular proteins as well, and different domains of protein along with the folds of protein. You also explained structural classification, and you also gave us the direction where we would study in the future as well in one of your organized lecture. And we are going to arrange this lecture as soon as possible, sir. So thank you again. And yes, we have with us two students from Sriji Science College, Balasinor, Sneha Patel and Parma Risita. Sneha and Isita, on behalf of Sri Govind Guru University, we welcome you, and we would like to have a review of both of you. That as a student, did you enjoy the lecture, or you have some suggestions? If you have, then you are kindly requested to provide your review, both of us. First, I would like to request Sneha. Sneha, અવાજ આવે છે મારો? If
Yes,
you
sir. Good noon, sir.
yes, if you can speak in English, it's good. Otherwise, you can continue in Gujarati as well. No issue. Yeah.
ओके सर. गुड नोट संकेत सर. 5 साल मैंने कंप्लीट आपके अंडर ही किया है. मैं एमएसयू की स्टूडेंट हूँ. अभी मैंने बीएड गुरु गोविंद से कंप्लीट किया है. हेलो.
હા, યસ, તમારો અવાજ આવે છે.
ઓકે સર,
કેવું લાગ્યું આજનું લેક્ચર?
મને તો કમ્પ્લીટ લાગ્યું અને સરની અંડરમાં હું ભણીને જ આ છે, તમને કોઈ પ્રોબ્લેમ નથી. એમની લેંગ્વેજ થી માંડી, પ્રેઝન્ટેશન થી માંડીને બધું જ અન્ડરસ્ટેન્ડિંગ છે. એટલે એમનો વે હજુ જે હતો એનો એ જ છે અને અમને પણ એની અંદર ફાવે છે.
Thank you, Sneha.
Yes, thank you, Sneha, for such a kind review. And please be connected with us. You can share this lecture to your friends as well, and tell them to be connected in NET GSet Life Science group that we have already created for the students who wish to crack different exams in life sciences. It can be PhD or MSc as well, or even NET and GSet, GATE, and all the other things that are taken by CSIR and ICR and all other boards of our Indian government. Isita, કેવું લાગ્યું તમને?
સમજાઈ ગયું બધું, એમ સારું લાગ્યું.
ઓકે, તમે કોઈ એક્ઝામ આપવા માંગો છો ભવિષ્યમાં?
હા, ગવર્મેન્ટની એક્ઝામ.
યસ. અત્યારે તમે કેવા છો? ટીવાય માઇક્રો કે બોટની?
બોટની
થર્ડ યરમાં છો?
પૂરું.
ઓકે,
GS
થર્ડ યર આ વર્ષે પૂરું કર્યું, રાઈટ?
હા હા
રાઈટ. ઓલ ધ બેસ્ટ ટુ બોથ ઓફ યુ, સ્નેહા એન્ડ
હા,
ઈશિતા.
થેન્ક યુ.
ફોર યસ. બીજું કઈ સજેશન હોય તો
હેલો
તમે
સર,
આપી શકો. જી
હું બાલાસિંધુની સ્ટુડન્ટ નથી. હું
જી
શ્રી
જી
કેએન પટેલ સાયન્સ કોલેજ કોઠાંબામાં
હા,
પ્રોફેસરમાં છું અત્યારે.
અચ્છા એવું છે.
હા
હા, એટલે ઓકે. તો તમે ફેકલ્ટી છો અત્યારે, તો અમે
હા
તમારું આમાં સ્ટુડન્ટ રિપ્રેઝેન્ટેટિવમાં નામ આવી ગયું હતું, પણ એ કદાચ મિસ્ટેક થી. પણ તમે અમારી સાથે જોડાયેલા રહ્યા અને ખૂબ જ આનંદ થયો કે તમે આટલી સરસ રીતે આખો લેક્ચર સાંભળ્યો. વી રિયલી એન્જોયડ યોર કંપની એન્ડ I would like to have you a request that you can join all your students to these classes because these are free and they are for students only. So those students who are not able to pay money for different expensive classes, they can join here and learn from this free online coaching. And faculties from all over the Gujarat and even outside, they are participating with us and they are giving their collaboration and contribution for the betterment of students. So thank you, Sneha madam, again for joining with us, and give our regards to all the faculties over there in Kothamba.
ઓકે સર.
જી, Ishita, આપનો પણ આભાર. Now I would like to
હા
have a vote of thanks from our today's one of the guest, Sahista madam. She is from Balasinor, Sriji Balasinor Science College. Sahista madam, you connected with us throughout the session, and definitely your vote of thanks and your review is special to us. Are you getting our voice, Sahista madam? Over to you.
સર, મારો અવાજ આવે છે સર?
Yes, આવે છે મેડમ. તમારો વિડિયો ઓન કરી દો પ્લીઝ.
હેલો.
યસ મેડમ, પ્લીઝ સે. તમારું ફેસ
હા,
થોડું નથી દેખાતું. થોડું, હા, ઓકે.
ગુડ આફ્ટરનૂન, નમસ્કાર. હું હટિયા સહિસ્તા અહેમદ, બોટની આસિસ્ટન્ટ પ્રોફેસર ફ્રોમ બાલાસિનોર કોલેજ. આજે એસડીજીયુ કોલેજ દ્વારા નેટ જીસેટ પ્રિપરેશન પ્રોગ્રામ ઇન લાઈફ સાયન્સ ઉપર વેબિનારનું આયોજન કરવામાં આવ્યું હતું, જેમાં MS યુનિવર્સિટીના આસિસ્ટન્ટ પ્રોફેસર ડૉક્ટર સંકેત ચાવલા સરનો હું આભાર વ્યક્ત કરું છું કે તેમણે સ્ટુડન્ટ્સને નેટ જીસેટ માટેનો જે અગત્યનો ટોપિક ડિફરન્ટ ટાઈપ્સ ઓફ પ્રોટીન્સ વિશે એમને માહિતી આપી. ખૂબ જ સરસ રીતે એમને પ્રેઝન્ટેશન આપ્યું, તે બદલ હું એમનો આભાર વ્યક્ત કરું છું. ખૂબ જ સરસ સર, તમને ખૂબ સારી રીતે સમજાવ્યું અમને. તે ઉપરાંત આ આખા વેબિનારનું સંચાલન કરનાર હું ડૉક્ટર રૂપેશ સરનો પણ આભાર વ્યક્ત કરું છું. અને આ વેબિનારની શરૂઆત કરનાર ડૉક્ટર અજય સોની સર, કે જેમને આપણે સૌને મોટિવેટ કર્યા. તે ઉપરાંત આપણા યુનિવર્સિટીના રજિસ્ટ્રાર સાહેબ ડૉક્ટર અનિલ સોલંકી તથા આપણા માનનીય વાઇસ ચાન્સેલર સર પ્રતાપસિંહ ચૌહાણ સરનો પણ હું આભાર વ્યક્ત કરું છું. તથા આખા વેબિનારની અંદર જોડાનાર આપણા વિદ્યાર્થીઓનો પણ હું આભાર માનું છું કે તેમણે શાંતિથી આખો વેબિનાર જોયો અને સમજ્યો. થેન્ક યુ સર.
Thank you, Sahista madam, for your very kind and enthusiastic words for all of us. and that definitely encourages us to work more and more for students and scientific community that we are representing. Thank you, madam, again, and hope you will be connected with us throughout the series whenever
Yes.
we invite you to join with us. So thank you again, madam. And with that, I think today we really enjoyed the lecture, and we completed our second lecture of third series, and totally we completed our 43rd lecture. So I would like to have brief information regarding upcoming lectures that we are going to organize in the next upcoming months. On the 1st of June, we have Dr. Minal Vamanya. Then we have Dr. Dehin Bhagat, then Dr. Jay Pandya, then Dr. Nilesh Kachadia as well. He is going to deliver from Indore. Then Dr. Eddie Kalola from Anand Agriculture University. Then Dr. Divakar, he is from Navsari Agriculture University. He will talk on proteomics. Then we will have some topics like cytology and even cyclin dependent kinases. It will be delivered by Dr. Jigna Tang from Saurashtra University. Then we will have different topics covered by Dr. P.V. Dharani and other microbiologists from different regions of Gujarat. So many more to come. And thanking you again, dear Sanket sir, for being alive and being connected with us. So we, on behalf of university, I would like to thank you again for providing your very important time to all of us and all the students who are watching us live. And I would also like to say that those who have not watched us live, you are kindly requested to go to the YouTube. You can see the whole lecture anytime, any suitable time, whenever you are free. So that is why we are organizing such lectures. So thank you all of you again. Have a great day. Bye bye all. Great evening to all of you. Thank you.
Thank
With that, I am ending this today's meeting. See you all in the next lecture. Yeah.
Thank you,
Bye
sir.
bye. Thank you.